Okay, so the last video was about the functions of the plasma membrane. That was the end of the plasma membrane. Now cytoplasm. First of all, A was cell wall, B was the plasma membrane, and C is the cytoplasm. This the term cytoplasm was it was the first discovered by the Dujardin, and he had named it sarcode. Purkinje had named it protoplasm. It has the following nature. It has colloidal nature, due to which is due to amino acids are suspended in the aqueous medium so as to match the density of organelles. This ensures that organelles remain suspended in the cell. The organelles will move only when cytoplasmic media moves, and the properties of colloidal suspension are following: tidal effect, Brownian movement, and cytoplasm. The two stages of cytoplasm. Okay, so tidal tyndall effect is important in plant cells for photosynthesis. Brownian motion helps in distribution of molecules in the cell. Cytoplasm exists in two cells which help in the which help in the better distribution of cytoplasm and better distribution of the chemicals. It is of the cytoplasm has two on the basis of density: cytogel and cytosol. Okay. More viscous is cytogel and less viscous is cytosol. All organelles and biochemical processes occur in cytosol, and in cytogel only it regulates the movement of solute and ions. Cytosol and cytogel exist in dynamic equilibrium, and cytosol to cytosol formation is called cytogelation, and gel to sol is called solation. Cyclosis is the term given by Amici, or it is also known as cytoplasmic streaming. It is the continuous and unidirectional movement of cytoplasm under a vacuole. It is again of two types: rotation and circulation. Rotation when the cell has one vacuole, and circulation when many vacuoles. So rotation is best observed in cells of young leaves of hydrilla, and the circulation is best observed in staminal hair of Trascandacia. Huh? Tradescantia. Significance of cyclosis is that it helps in the distribution of gas, solute, and food. It helps in the removal of waste. It helps to maintain the temperature of cell. How it maintains? Because rate of cyclosis is is directly proportional to temperature. The more is the rate of cyclosis, more the movement or circulation or rotation, more will the temperature. If a cell is exposed to high temperature, its rate of continuity increases, therefore distributing the additional temperature. That means if temperature in the cell is high, so rotation will occur, and the temperature at single part would eventually spread to the whole cell. Okay, and thus decreasing the additional temperature. Therefore, net gain or increase in temperature is minimized. Now, cell organs, that is D, fourth part. Endomembranous system, okay, group of four organelles connected indirectly by function. These are the main organelle of the cell structural. Okay, it has four living organs: endoplasmic reticulum, Golgi bodies, lysosome, vacuole. Okay, endoplasmic reticulum was discovered by Thomson and Potter. It is the network of membranes spread throughout the cell. So it is that is connected to both nuclear envelope and cell membrane. This is a diagram of endoplasmic reticulum. This is the nucleus. These are the ribosomes on the cisternae. Here are tubules, as well as you can see the vesicles are lying around in the cytoplasm. Towards the nucleus we have cisternae, and tubules are far from the nucleus. Okay, cisternae are near to the nucleus. It has three parts: endoplasmic reticulum, cisternae, tubules, and vesicles. Cisternae are those structures which are continuous around the nucleus. They always have ribosomes. Always have ribosomes. The membrane of the cisternae are associated with 60 subunit of complete ribosomes, that is large subunit of ribosomes. We know that 60 is for large and 40 is for small. So it has 60s, and then now tubules are the tube-like or branched or unbranched, usually free of ribosomes. Very rarely have them. Spread throughout the cytoplasm. Vesicles are spherical bags, always free of ribosomes. Vesicles never have ribosomes. Cisternae always have ribosomes. Tubules may or may not. Okay, vesicles are randomly diffused in cytoplasm, as said earlier. Cisternae, tubules, and vesicles are interchangeable. That is, they may change from this one to this, tubule may become cisternae, and vesicle may become tubule. Okay, they have very similar membrane, and the matrix is also similar in these three. There's a structure. Okay, types of endoplasmic RER, SER. RER due to attachment ribosome. Cisternae will be near the nucleus, and SER that is having smooth, which has very less or absolutely no ribosomes, that is tubules and vesicles in actual are away from nucleus. Okay, in SCR, membrane has special glycoproteins called riboforin 1 and riboforin 2. These are very important. Okay, next part would be about this. It requires a lot of understanding. RER can be changed to SCR by removing riboforin, but SER can never become RER because of absence of riboforin. Okay, riboforin 1 and 2 are special glycoprotein which help ribosome to attach with endoplasmic reticulum membrane. This is the small subunit, this is the mRNA, and this is the large subunit which is 60s 40s, and here is groove, that is empty space, and a polypeptide comes in through this mRNA. Okay, it extends from there, and this whole ribosome and mRNA structure is connected to the lumen of the LCR through the riboforins. Okay, large subunit of ribosome has groove for the newly synthesized polypeptide. Okay, large subunit of ribosome attaches to the membrane of RER with the help of riboforins, so that polypeptide directly enters into the lumen of endoplasmic reticulum. The ribosomal subunits associated with the mRNA to initiate polypeptide synthesis. Okay, the first step is polypeptide synthesis, and such polypeptides usually have a short peptide chain which acts as a signal peptide to a defined riboforms. Now, that is the mRNA and the two ribosomes subunits. Okay, 40S and 60S. Signal peptide has formed, and it will search for the signals for riboforins of endoplasmic to attach to. Okay, the ribosome which is attached to ER now. Okay, now it has attached to the ER. Okay, the signal peptide is found and it is attached. Okay, will have its groove. Now groove would be formed inside it. Okay, and align that in such a way that polypeptide enters into the lumen of ER. Now the lumen into the lumen. Okay, polypeptide will enter. The signal peptide will now be separated from polypeptide. Okay, and this will be called the cleavage of the signal peptide. Now there would be no more signal peptide. Third step would be, as polypeptide grows, it associates with the glyco group, forming a glycoprotein. That is, the PP would rise and rise and rise and form glycoprotein. Okay, the function of glyco group is to provide orientation of polypeptide, thus giving it towards guiding it towards the functional site. After completion of translation, the two subunits of ribosome and mRNA will be removed. 60th subunit may remain attached to ER for a long time until it is needed. That is, the bigger one would be still attached to the endoplasmic reticulum, and the smaller one and the mRNA would go back, would separate. Now, functions of ER, that is both SCR and RER, it will provide surface area for membrane associated function, provide some support to the cytoplasm. Functions of RER, it is the site of it is the site of protein synthesis, that is lysosomal protein, secretory protein, membrane protein. Neuron has Nissel's granules that is made up of RER and ribosomes, and that helps in the synthesis of neurotransmitters. It provides surface area for attachment of ribosome. It will form new nuclear envelope at time of cell division. Functions of SER are following. It helps in the synthesis of lipids, steroids. Okay, it is the precursor of spherosomes. Okay, in muscle cells, it is also called as sarcoplasmic reticulum. It maintains the calcium ion balance. In retinal cells, the SCR form myeloid bodies. Okay, which is present in the eye pigments of vision for vision. In liver cells, they help in the detoxification of drugs, alcohol, and narcotic drugs. In our liver cells, these are present for glycogen metabolism, means glucose to glycogen, and vice versa. It is also present in the desmotubule of plasmol desmata. That was the we studied it. New cell membrane is also formed by SER. Endoplasmic reticulum is not well developed in dividing cells. Okay, by the centrifugation, ER is isolated as small vesicles called microsomes.